If you're ready, we can start. Are you ready?
Yep.
Okay. Welcome to another statistical seminars. It's my pleasure to introduce Dr. Gao Wong, who is an assistant professor of neurological sciences at Columbia University. Dr. Wong got his PhD degree from Baylor College of Medicine. Dr. Wong's research focused on the study of genetic etiology of neurological disorders through integrating from diverse human populations large-scale multi-omics datasets, as well as phenotypic records such as self-reported phenotypes, electronic health records, and medical images. Today, he's going to share with us his recent work on testing 15 billion eQTL association. Let's welcome our speaker.
Thank you, Yang, and thanks for everyone for coming here virtually. And it's a pity we couldn't meet in person this year. But as Yang was introducing me, mostly as a computational biologist, we are mostly interested, in lots of practical applications, especially in the Department of Neurology at Columbia. We are mostly interested in all aspects, genomic aspects about the neurodegenerative disorders. But today's talk is actually mostly my recent work as a postdoc with Matthew Stephens at University of Chicago, and where we were actually still motivated by practical problems. As you can see there, it's a testing association between genetic variation and expression, which is called the eQTLs. And it was my goal to develop the methodology under this context and apply that to a more diverse context in our Department of Neurology. And so today's talk, I'm going to first discuss mostly on the methodological aspects of this association testing motivated by this eQTL problem. And then at the end, well, I'll talk about the future applications in neurology, because like I said, I really just started and mostly been working on proposals and pre-processing of data. We don't have any neurological results to show for today. Okay, good. All right, so the first thing I want to discuss is the multiple testing problem in the context of genomics. So the background of, well, I guess people here are familiar with multiple testing, but in the context of genetic association study, we're talking about a specific type of study design where we simply say, well, a simple design would be take a population of samples, say you do genome sequencing on thousands of individuals, and then you measure their phenotype as well. For example, you ask a person, or from medical record, if this person has type 2 diabetes, and some person don't have type 2 diabetes. And what you do is that you group people into the ones with type 2 diabetes as cases and the one do not have type 2 diabetes as non-case or controls. And you simply compare for each of the genomic variant in the genome, if there is an enrichment of a particular mutation or a particular form of genetic variant, which we call an allele, in the cases versus controls. And then if there is an enrichment, then we call that this variant is associated with the disease. So this is roughly like the genetic association studies. And the multiple testing thing coming to play here, as you can see, is that for one disease, like type 2 diabetes and not, then you can test for, there are 3 billion base pairs in the human genome. And for the ones that are mutatable, let's say, there are still millions of them. So for one disease, you've got to test for millions of association analysis. And actually, this was in the context of disease genotype association, in a context maybe even less familiar to non-biologists, but still of great interest is the context of studying the association between a gene expression and the genetic variables across many human tissues. So this is like a sketch of the type of problems we are trying to tackle, or the type of problem that actually motivated methods development here. So in this is a bubble plot just showing you some association testing results. And each of this bubble here, the size and the color measure some estimates above that association. In this case, it's actually some type of a log p-value, a minus 10 log minus log 10 p-values, and the size is like the effect size of the estimate. What's important to look here is that, so on each of the column, it's one variant, it's one genetic variant. So those are all the IDs for the genetic variants. And on each of this row, it's actually a human tissue. So for example, brains, heart, blood, skin, so on and so forth. And we are looking at this one particular gene, the expression of this one particular gene. So what we do is that we take measurement of the gene expression for this ACTN3 in, for example, this tissue, I remember was stomach tissue or something like that. And then take this tissue and measure the gene expression, and then do this association testing, type of that enrichment thing I was referring to earlier. So try to correlate the mutation status on each of the SNP with the expression. So we end up having lots of these tests. And just by looking at this picture, you can see that, so first of all, there's many tests, just even for a small gene region across multiple tissues. And you can also see that many of these tests seems to be quite similar. Not just these rows are similar, right? So if you look at this row and each of these columns, they look kind of similar to each other, right? But also the patterns across all these different columns are also show some similarity. For example, here it's more sparse and here it's more dense. And you see like some cases it's very sparse. So there are a lot of tests here, and there are a lot of similarity going on in this thing. So our task is actually, this kind of data was from the so-called genotype tissue expression project, where we were dealing with 50 different tissues on 3,000 genes. And each of them we're testing 10,000 so-called cis SNPs. Cis means, nearby. SNPs is a single nucleotide polymorphism, which is the genetic variants. So we're doing, this is like 10,000 SNPs per gene for the test. So we end up having these 15 billion tests. And if you click on this page, it's essentially that bubble plot I was showing. As you can see, there are some patterns going on there. So, okay, well, if we take a step back and thinking, we are actually doing lots of tests. So we have to pay for, the multiple testing. Is that a terrific burden for, doing just so many tests? And in fact, in association studies, if we still look at the old p-value approach, what people have to call significance for that has to have a p-value of 10 to the minus 8. It's not like the 0.05 usual thing. They kind of have to correct for all those testings and use a very stringent p-value. So the more tests it seems, the more penalties we have to pay. But is that really the case? Just because of the structures and correlations and similarities we seem to be observing in the data, what we claim is that the more tests we have, the more opportunities we have to learn. Because, maybe those similarities and structures in the data can actually help instead of penalizing the, our statistical procedures. So we call, instead of a multiple testing burden, a multiple testing party. And that was, initially from Joe Pickrell's blog post, and became very popular, at least, among many people. Like our group back in Chicago is like, multiple testing burden is, this phrase is forbidden on our floor. We have to use multiple testing party instead. So of course, to enjoy this party is, my goal of, to invite you to this party is my goal of today's talk. And I'm going to discuss two separate type of analysis, and finally, combination of them to actually integrate everything on the bubble plot I was showing earlier. So the first one is a new Bayesian variable selection method. And this is trying to deal with the problem of, having, so if you're just focusing on this example, on the expression in stomach, and then look at the genetic variations controlling for the expression in stomach, and what we can see is that, there seems to be some signals going on, right? So these are like very big dots signifying for signals. And then there's a decay around these signals. Sorry. But then the problem is that actually we know that many of these variants are highly correlated. So if you're only analyzing one variant at a time, as in this case, you end up having lots of correlated tests, but the effects are here are really sparse. So this fits nicely into a variable selection problem that many people should be familiar with. So it's like a conventional regression setup where you have this y equals xb plus an e, and then the assumption here is that the effects are sparse, but the observation, the b hats are actually dense because many of these axes are correlated. So the task for variable selection is, of course, to identify the true non-zero effects when these axes, the data, are very correlated. So this seems to be like conventional setup, but in genetics, it has some quantities that we really want to measure. We just don't want to select some variables. To be specific, let's consider, for example, just the two variants that has non-zero effect for disease, and this is like a large genomic region. I'm drawing a chromosome, but doesn't have to be this large. So maybe we're looking at maybe like 10,000 genetic variables, and only 2 of them have a non-zero effect on a phenotype, for example, disease or a gene expression, right? But then for some of the other variants, which we call the SNPs, for some of the other SNPs, they look exactly identical to the ones that are truly have a non-zero effect. But just because they are completely correlated, their genotype will look exactly identical, and their observed effect size will also be exactly the same. But then to a biologist, when you report these 2 SNPs, you get exactly the same univariate effect size estimate. And to a biologist, they're really interested in knowing, which one is actually in red. And in this case, just from the data, it's impossible to know which one is in red. So what the biologists want to know is something like, can you tell me all the possible collection, the set of the SNPs, and in which, there's both the effect variable and the ones that you really cannot tease apart. So we are saying that, for example, this type of inference is different when you do some type of a lasso variable selection, which actually I simulate a very small data set just under this model, just by using 4 variables, and 2 of them have a non-zero effect, but the other 2 are identical to these variables. And for example, if you run lasso, then you can kind of see what could happen is that it will pick up some of the variables, like this red one are the true zero effects. But then because of the other one, it's also identical to some non-zero, to some variables that doesn't have an effect. So what lasso can do is that in that case, it randomly just pick up the other one that doesn't have effect and dropping the one that actually has an effect. So this is some properties that, in our context, it's not desirable, because if you want to do a prediction for the phenotype, probably that's fine. You can predict either using this one or that one. But if you want to, actually understand what these features are, especially in biology, this feature could be a functional genomic variant, and this other one could be just a neutral variant. So you do want to understand, you don't want to get rid of this one. And then that's why the Bayesian variable selection regression are very popular in our field of study. Actually, it's kind of the expertise of one of your faculty, Chen Zhou, and he does lots of work on this. Those are very influential contributions. So, but roughly speaking, what the Bayesian variable selection regression work, how it works is that it try to kind of sample all the possible combinations of non-zero effects, and then come up with, for example, in this case, you're sampling like 4, and if you're assuming, for example, 2 of them are non-zero, have non-zero effect, then you can come up with these 4 different possible combinations of where the non-zero effects are. And then you do some type of sampling, or you work that out one way or another for these distributions, and you marginalize that, and you get the probability for each of them. And this is actually a nice inference, because first you can see that this is the posterior inclusion probability. You can see that every variable in question have 50% inclusion probability, which is true because we know that we can't tell between these two variables and we can't tell between these two variables. So these two variables kind of share the 100% probability, and so do these two variables. But then it really doesn't. So when you look at the result, then it really doesn't answer this question though, because it doesn't really give you back this result automatically. Because in here, the inference should be, either this one or this one is non-zero, and either this one or this one is non-zero, right? So we are talking about this type of statement, and a conventional Bayesian sparse regression just doesn't automatically give you that interpretation. So that's one motivation why we want to bother with developing a new variable selection approach. And the other one is probably well known, is that many of these sparse regression, Bayesian sparse regression, use some type of sampling, are very computationally intensive. And when you are working on problem like on the whole genome scale, it's just very difficult to implement. And in our case, we are talking about tens of thousands of individuals for our neurological study. But then if you imagine like the biobank type of data where you have like 500,000 samples and you have genome sequencing, so it's not just 10,000 variables, it could be 100,000 variables. So that's why the conventional Bayesian sparse regression also has computational issues. And but just to focus on the type of inference we can provide, so this is the result of our new approach called the sum of single effect model. So in this case, not only do we make an inference for the posterior inclusion probability, which is identical to a regular Bayesian sparse regression, but also we can automatically give you some credible set. And this 95% credible set has a definition of 95% probability to capture exactly one non-zero effect. So these two in green belongs to one credible set, and these two in red belongs to another credible set. So it gave you that exactly that statement. And we argue that without additional data, this is the most you can learn from this from the data you have. And this is already very interesting because for biologists, they can really focus on like one variable at a time instead of trying to figure out what kind of combination are there in this whole site it's reporting that they can do some functional validations on. So to briefly outline how our sum of single effect model work, still consider the conventional Bayesian sparse regression, but instead of writing it as a spike slipe type of prior for the Bayesian sparse regression, we instead formulate the effect size into a sum of independent single effect size. So this is like showing in this cartoon where, for example, three of these variables have non-zero effect, and then we factorize that somehow into a summation of three single effects, and each of them only have one effect variable. And that was actually justified by a variational approximation to the posterior distribution. So when you have that full posterior, we assume this mean field variational approximation by using a product of the posterior of the single effect model. And importantly is that even though we fully factorize the single effects, and making the single effects independent, but the constraint here is that within each of these effects here, we don't factorize over these elements. So we are factorizing at the single effect level, but not at variable level. So if you see the variable level, it's usually like B1 to BP, n times P type of. So we don't factorize B1 to BP. Instead, we use some very stringent assumption on this. So this assumption essentially says that the effects themselves are independent, but their observations can be similar. So this is this at least match the context of genetic fine mapping and in quantitative genetics, this is an accepted assumption. And so our paper tries to show, justify this variational approximation to this true posterior. And we also provided a algorithm to easily implement this variational approach. And an outcome of that algorithm is that it can naturally report for each single effect, some single effect level credible sets. So our algorithm, I'll show you right away after these two data demonstrations. But so maybe we can look at here for one concrete example. So here is still a simulated association study where I just randomly pick up two variables to be the non-effect variable and generate the data under this model, the regular sparse model. But just because this axis, the variables are highly correlated because I took them from the real genotype. So the real genotype of 600 European samples. So because of the genotypes are correlated, it happens to be, this particular genotype is correlated with this genotype and also correlated with this genotype. As a result, it kind of, gets some signal. If you do univariate analysis, it kind of gets some signal contributed by this one and contributed by the other one. And in biology, a lot of times, when we see lots of correlated variables, it's just unfortunate people make the assumption that, the signal must be here and everything else is because of some type of correlation with this signal. So they call that the top signal or top variant approach. But in this case, if you take the top variant, it's going to be very wrong. And from our analysis, we're able to capture not only these two variables, but also, a credible set for them. And for this one in biology, we say, so biologists will be very happy with this one because, it narrows it down to 3 variants. And one of them actually has a very high posterior inclusion probability. It means that this is probably a good thing to follow up, even though, marginally it doesn't have as a strong p-value as in here. But then for this one, as you can see, it has a lot of variables, but their posterior inclusion probability are all very low. And although, the red one are like the relatively high posterior inclusion probability ones in this set, still it's all under 20%. And the reason is because, in this set, it has a 37 SNPs with a minimal absolute pairwise correlation, 97%. So this is very high correlation. So it's very difficult to tell them from each other. But our method is going to tell you, this is something I can't tell, but I can tell you the collection of the 37 variants has one of the non-zero effect. And this also gives you a feeling of, the type of data we have in genomics is that they're really highly correlated. And this is a demonstration of how the algorithm works. It's actually, we call that iterative stepwise Bayesian variable selection. So what it does is that it simply is kind of like a conditional regression approach where you, for example, as you first regress out the most obvious signal, like the one in here, and then you see what signal are remaining in the data. You regress out the other one, and you regress out the third one, go so on and so forth. And this is actually just this conditional regression, but done in a Bayesian analysis framework. But the key is that we just don't run one iteration, we run more than one iteration. So if you see the second iteration, things starts to change, right? So you can see the top signal is kind of weakened, but something else is coming up. And if you keep doing that, and you can see that as the algorithm goes on, we are able to kind of escape the initial trap of the incorrect inference from the top signal. And if you keep going, keep doing it, so you can see that one eventually disappear. And I think the nice thing with a model-based approach, well, this is an algorithm, but the nice thing with the model is that we can actually analytically work out the evidence lower bound, kind of like a marginal likelihood, and we can therefore have a very good stopping criteria. Versus if you're only using, for example, relying on just an algorithm. So what happens is that from iteration 9 here to 10, for example, you see the variables, the inference parameters, they don't really change too much. So maybe if it's an algorithm, we call, oh, so the inference don't change too much, we stop right here. But if we have a model-based approach, then we can always check the increments of the variational lower bound. And then that tells us we should keep going. We should just keep going. And then there, we stop. So we have a very nice stopping criteria for this algorithm. And just some empirical studies showing the utility of this approach is that we are combining this with a bunch of the Bayesian variable selection methods for fine mapping, and our method SUSY outperform them. I think part of the reason is because we are essentially working on the same problem setup, so we don't really expect our method to be particularly powerful than the others. Although the credible set is a contribution, but I think the power actually comes into the fact that we are using a empirical based approach where we actually estimate some of the prior parameter using data from across many regions. So that's why we can have a better prior versus for the others, they have to make some assumptions on the priors and they work that out internally. So here with the empirical based approach and with just lots of data that we have, so we kind of turn the multiple testing penalty into a blessing because we can learn from all these tests and have a good estimate of the data of the hyperparameters. And also this algorithm is much faster than these many other tools, even though it's just implementing this loop in R, it's already very usable. And in fact, a lot of people have been applying this to large scale association studies. So that was for the variable selection in the univariate case.
I have a question about the comparison between the methods. So here, some Bayesian methods are compared with your method, but I just thought that some frequentist method can also be compared because the lasso has that problem of selecting just one variable out of some correlated variables. But as far as I know, the elastic net can address that kind of issue to some degree. So I thought because frequentist approach may be much faster than the Bayesian approach, so I thought that can also be compared.
Yes, so actually we have thought of, actually one of the reviewer asked us to compare with, elastic net or something like that. But one thing is that we are actually dealing with very different problems or type of inference. So I guess the most important reason is that they don't really provide a posterior inclusion probability, but we can potentially use some of the rank, for example, the rank of the SNPs to compare. But then this is just the one comparison we're making. And for credible sets, it's really difficult to compare with some of the other approaches that doesn't really provide credible sets. And it's also, because of the ability to make a posterior inclusion probability type of inference, the results are much more digestible to our biological applications. So I guess that's also another reason why all these so-called genetic fine mapping methods, they never actually look into using lasso or elastic net, the type of the frequentist methods.
Yeah, thank you.
Okay. So for the multivariate analysis, if we, instead of focusing on all the variables, let's just look at one variable at a time. But this time we're looking across all the different tissues, right? So you can see kind of lots of similarities going on here, but from here you can see, oh, so this variable seems to be impacting all of these tissues except for one of them. And that's very interesting because why biologically it doesn't impact this particular one tissue, right? But if you look at the sample size in here, it's only 120, so maybe it's just a lack of power. And in fact, a reasonable approach should be able to kind of impute also, because it's impacting everything else, and maybe lots of the QTLs actually do impact across the board all the tissues, and it's really rare to see something doesn't impact one tissue but impact everything else. So it should be able to kind of impute this and realize that's an artifact. So that was the type of multivariate analysis we want to be able to do, because we want to leverage information from similar conditions and to inform the ones that doesn't really have a good statistical power. So that boils down to actually how similar these things can be. And so if we are talking about the context of meta-analysis, so for example, we make assumptions on, say, fixed effect or variable effect meta-analysis, we can, it's just a measure of, you know, how similar the effect size can be. But in here, we have some much more complex situation than simply assuming fixed or random effects. Because, for example, if you look at many of the brain tissues, and you can imagine, you know, their effect size are actually more shared than between a brain tissue and a heart tissue. And for the heart tissue, their effects are probably also shared. But then between tissues, there are some type of sharing, but not too much, right? But of course, you know, there could be some of the variables that have an effect across the board, but independent from each other. Also, some of the variables may have very tissue-specific effect. Means that the variable can change gene regulation in brains, but it can't change gene regulation in heart or everywhere else. So how do we, so recognizing all these possible patterns of sharing going on in the data, how do we leverage the information? So first step is, of course, to try to identify the possible patterns of sharing. So we just do a simple factor analysis as a demonstration. Here, well, in practice, we did something more than that, but this is just a demonstration showing it does exist. So we just assume a low rank plus a diagonal structure for the matrix factorization. And then we end up having a few factors that you can see. So this factor showing an effect across the board, but the effect size are heterogeneous. And this second factor captures somewhat like a brain specific effect, as you can see in yellow. So those are 9 different brain tissues. And for some of the other things, it captures something from whole blood or something from skin, like sun-exposed skin versus non-sun-exposed skin. So it's kind of, you know, you can imagine doing some type of analysis to decompose your data into a combination of all these different patterns. So based on this result, we propose a framework called the Multivariate Adaptive Shrinkage model, where this is a conventional multivariate model for the true effect size and assuming the known standard error. And that's the observation from one variable, but cross conditions. So this is like an R-dimensional vector. And in this prior setup, instead of just using one multivariate prior, we are actually using a mixture of multivariate priors. With each of these things, this effect size is exactly reflecting the structure or the possible structure, the structure we propose or the possible structures we identify from data. But then we actually estimate from the data the contribution of each of these structure. So even though, you know, both of these quantities are learned from the data, you know, we do it two-step. We first get all the possible pattern of sharing in here, but then we assess from the entire data set what their contribution should be like. And that was actually a very straightforward, because, you know, the only variable that we don't know is this weights. And that turns out to be a convex optimization problem, because initially for a mixed model, we have to do some EM algorithms, but then realizing, you know, this is a simpler problem. So it's very fast to also estimate these numbers. So once we have these numbers, so again, that's another you know, proof of what we were saying. The multiple testing is an opportunity to learn, because not only do we learn the possible pattern of sharing, but also from the entire data we learn the weights. And the more the test, the better actually we can learn from it. And by applying, so then we have an estimate of these pi's and then those UKs. So we complete this model, and you can understand this as a kind of a meta-analysis model, but with a flexible mixed prior setup. And applying that model to the GTEx data we have for one variable, what you can see is something like this. These are the original estimates for the betas and the standard errors plotted here. And you can see it's kind of noisy, and lots of them are you know, they overlap with zeros. But after applying this method, you can see that the observations are a lot less noisier, and the effect size estimates actually kind of get shifted a little bit, but it's more confident. And then you can see a clear pattern of positive effects, but smaller effect in non-brain tissues, large effect in brain tissues. Versus here, you do see a bunch of these negative effects that you can't call significant or not. And in some of the other cases here, it's very noisy. It seems like lots of the single univariate effects like this one is significant in this particular tissue, right? But then because of our prior kind of regulates it, and we end up having this result, identifying this is a truly tissue-specific effect. Well, not truly, but from our model, this is tissue-specific, and many of the effects are shrunk to 0. And because we have a proper framework to actually quantifying the scale of the effect size, we can do something better than some of the other approach. Because some of the other approach can say, this is an effect across tissue or specific to tissue, but it can't give you the estimate of the effect size. So that's why we're saying it's a plus using this framework, because it reviews what we call the quantitative heterogeneity between the tissues. In other words, so if we look at the effect size from the tissues and simply posterior effect size from our estimate and simply make a plot, a heat map, and you can see that it reviews, these interesting patterns. Like for example, these two brain regions and these two brain regions are clustered together, and these are of course controlling the motion, and these are mostly many other functionalities. So and also you can see that for the skin tissues, they are nicely clustered, and heart tissues clustered. And you can see many of these observations are consistent with what we know already about the biology. But there's something also you can see that indeed the brain and non-brain don't have, well, they still have some type of correlation, but not as much as between these different tissues. So that's one contribution that our model can provide for biology. And again, then bear in mind that here we are only dealing with the columns, right? So this is still looking at one variable at a time. But then these variables can also, across the different genetic variables, can be correlated, as I was describing in the first section of the fine mapping problem. So ultimately, what we want to do is we want to be able to reanalyze this whole bubble plot and plotting different effect sizes, but then taking into the consideration the fact that many of these are correlated. So along this line should be sparse, and many of these tissues are shared. So along this line, it should be, well, maybe shared, but also or maybe specific. So along this line, we say, should be more like regulated and less of these outlier cases. So that's why we're trying to combine these two approaches to actually doing this variable selection across multiple conditions. This is still ongoing work. And another motivation, actually more biological motivation for this study, is that if we don't do this type of study, what we can see sometimes in our data is that one variable actually contributes to an increased expression in heart tissue, and the other variable contributes to the same variable seems to contribute to a reduced expression in brain tissue. And that's really some weird biology, right? So why does it enhance the heart expression but suppress the brain expression? But if you think about possible artifact, this is possibly what's going on, is that this observation is a result of two different SNPs, different genetic variables. But just because of one of them is in negative correlation with the other one, so the observation, so both of these are surprising the expression, for example, but just because of the complete negative correlation between this and the other one, so this one will appear to enhance the expression. So that's an artifact, and we do see that a lot. I mean, we do see this a lot in the data. But without some type of analysis, like what we are about to discuss, it is really difficult to tease that apart into something, into what might really be going on. So the first step, of course, is to generalize our univariate fine mapping model, the SUSY model, into a genome-wide approach. So this is actually outlining the structure of the SUSY model, but the first part is exactly the same as our SUSY model originally proposed. We're just swapping the prior from a normal distribution to a mixed normal distribution. So we can learn better the possible distribution. We don't have to really assume normal across the genome. And then these estimates are performed by combining multiple genomic regions. So there we have a better estimate of this a mixed approximation. And we know that this seems to work really well for our multivariate case. So we then just rewrite this into a multivariate notation. And in this case, we still estimate across the entire genome the mixture component, the patterns of the mixture component, and the weights of the mixture component. But then we add additional parameter to control for the scale. So that's some technical detail because we want to be able to account for both the large and smaller effects. So we just use some patterns here and add it to a scale factor, and this scaling factor will be estimated in the variational updates. So this is what we call the multivariate SUSY approach with adaptive shrinkage, and we call that MV-SUSY. But of course, you don't have to really be using a mixture prior here because many applications, if it's not genomic application, it doesn't have a mixture prior. You can still throw in whatever prior, or just for convenience, one single component, regular multivariate prior. But the key for the genomic study or relevant to the multiple testing scheme is that we are able to leverage information from the genome. So we can take this empirical based approach very much justified. And there are actually not very many methods out there to perform the multivariate fine mapping. And the only one that we can find that actually work reasonably well just doesn't perform as good as ours. This is a comparison. We don't even have to estimate the variables from the data. We can just throw in a bunch of pre-specified configurations, and we can outperform the existing method. And in terms of speed, because the other one is also running this very computational intensive MCMC. So we are still this variational approach in the multivariate case. So we are still very fast. And does it actually answer the question of the genetic architecture I was proposing? If we observe this one variant having opposite effect in two groups of tissues, is that really an opposite effect, or as we assumed, it's coming from two independent signals? So running our multivariate fine mapping approach, we are actually able to identify two clusters of variants in the form of credible sets. If we look at the top in each of these credible sets, they are actually very much correlated. So that makes it difficult. So that explains this phenomenon, where if you look at just one of them, it's going to show marginally across the board, just looking like this. And if you plot the effect size, then you can see that these two credible sets are nicely teased apart, and each of them represent a group of effect corresponding to a specific type of tissues. Okay, so that's so much about my the previous work of analyzing the 15 billion QTLs. Now we are currently still applying this method to the GTEx data. And we are actually having some challenges there because in GTEx data, there's a lot of missing observations for the phenotypes. For example, a person enrolled in the study don't have their brain data available, right? They only have their blood or skin tissues. Some of those data are missing, and we worked out a version that can handle this missing data in this framework. So we're still applying this to the large scale 15 billion associated testing problem. Hopefully we can have some results soon. But like I said, I am now in this biological or medical department, and we have lots of data that are beyond, just analyzing the eQTLs. So the eQTLs, the data we can assemble here is actually has 2,000 brain samples. So those are like after we follow the cohorts from European and from Hispanic ancestries. And after they pass away, well, of course, for cohorts that we record everything they have, right? So, but after they pass away, we collect their brains and we do all sorts of multi-omics assay and get this data. And because, Alzheimer's disease is really a prevalent disease in the old people. So many of those samples that we have are actually Alzheimer's disease samples. Actually, we have a bias of collecting for Alzheimer's disease. So we end up having 700 of these brain samples processed so far are actually suffering from Alzheimer's disease. And then we have independently another 40,000 Alzheimer's disease samples from different populations, but they have the whole genome sequence data. And one particular thing about that data set is that, there's population data, there's also family-based study design, where you can see clearly, the Alzheimer's disease, you have the genotype data for both the parents and for the offspring, and then the parents have Alzheimer's disease and the offspring also has it. So you can imagine maybe a particular genomic variant is enriched in a family. So it could be a more powerful study design for some of the variants than in population study design. And with all this data, we are actually generalizing the existing framework into multiple directions. So one thing is when you see my first few slides where two variables are exactly identical, like 100% correlated, just from within the data, you don't get to know which one is truly, has a functional impact. But if you have also the multi-omics data, for example, if you see some of those fall into a promoter region or a transcription binding factor region, like all those functional genomic regions, versus something fall into what we conventionally call a junk region of the genome, then you can say, even though those are 100% correlated, then the one with some functions is more likely to be a true variable. And in order to do that in a very principled framework, of course, that can be formulated as a type of prior inclusion probability. And we just have to incorporate thousands of those annotations and different genomic features. And they can come in different forms, like, then they can be QTLs by themselves, or they can be like region-based annotation. I was mentioning, a region is a transcription binding factor. And we do some simulation studies for this, and we can see that if you use some of the annotations, then the top SNP that you identify in credible set are the true causal, true non-zero effect SNP probability become much higher than if you don't use that information. So we are still working on trying to incorporate thousands of those and do it whole genome-wide efficiently. And another thing is when we have so many variants there, another interesting thing is that we want to be able to co-localize. For example, we see some of the variants changing expression, and we also see some of the variants changing methylation and histone and of course, many of the variants, when we do genome-wide association study, they seems to be correlated with the Alzheimer's disease status. So if you consolidate all this evidence together, so this is actually just a fun mapping in each of these traits for a well-known Alzheimer's disease gene. So this is the genome-wide association studies for Alzheimer's disease. And these are results from what I was describing, the brain samples. And you can see that there seems to be some overlapping signal in here. We don't have a very nicely typed signal for histone methylation, but something seems to be here. And then elsewhere though, there are quite a few things that are regulating methylation and histone methylation, but they don't seem to have any impact on the expression or on the disease phenotype. So, but you can see that just by putting together a bunch of these fun mapping results for different data types like this, it's kind of still noisy. For example, this part, maybe there's something going on, that we can, actually integrate this and perform some formal colocalization type of analysis. So that will be a very interesting thing to understand how the variants work for the disease at molecular levels. And finally, we are generalizing this and analyzing functional data and then try to define a new type of methylation QTL. Because right now you measure methylation for each of the base pair, we call that the CpG site, but methylation usually happens like in a unit of a region. So this describes the problem as a kind of a functional data because there's some spatial correlation between the methylation sites. And in order to fine mapping this functional data, we can come up with some type of a wavelet transformation and use apply the fine mapping methods in a transform space. And of course, you have to design the corresponding prior mixture and the corresponding kind of algorithm for analyzing that, because there the scale of the conditions is much larger than the scale that we can typically analyze for like the actual phenotypes, because the methylation region can be very large. So that's all of that. And a reason I was trying to introduce our data is also, if some of you are graduating and looking for postdoc position, we have a nice environment here at Columbia, where it's an expertise of computational biology, statistical genetics, and combined with neurological sciences. So we can, if you're interested in real data applications, we welcome your contribution. Okay.
All right, okay. Oh, thank you very much for the very interesting talk. And it's time for questions. Do we have any questions for the speaker?
Thank you very much for the talk. So returning to your title, testing 15 billion eQTL associations, then could you please say something on the practical aspects of your algorithm? Say, maybe can you run your algorithm in parallel, or do you say chop your data set into different different parts and then run your algorithm on each part separately? Things like that. Yeah, thanks.
Good. So yeah, well, I guess, yeah, this figure kind of summarizes what in practice what happens. So what we can do is that because we are taking this empirical based approach, and so what we do is we only focus on the signal part when we are estimating this effect size correlation. So to discover the pattern of association, we don't actually use 15 billion results. What we do is that we take a subset of the results from the 50 billion, and we can make sure those are independent, because we can just select the variables that are far from each other. And we say that, that's representative of the rest going on in 15 billion. So when building the model, we are not running that in parallel for estimating some of the hyperparameters. But then with this hyperparameter fixed, what we do is that we can chop these 15 billion regions into units of genes. Because here the focus again was to testing 16 billion actually cis eQTLs, because we are only interested in the QTLs that are nearby a gene that regulates it. So then there we can chop that into 30 different units and try to apply this model on 30,000 different units, and only estimating the effect size parameter specifically for each of the gene. And the idea here is still, leveraging information across the genome to kind of shrink. So that's our shrinkage prior. So that part was done on a subset of data. So computationally, we should be fine, and it's scalable because after you learn across the board the shrinkage parameters, you are down to a manageable task involving one gene, 50 different conditions, and only 10,000 variables.
Thank you.
Thanks.
Any other questions? Well, I have a quick question related to trans questions. How do you actually estimate use of K, the correlation matrix?
So here, we can use some matrix factorization. Actually, one simple approach would be just using PCA, right? We can just run PCA and we can get those principal component as a variance covariance matrix. But the other nice thing is that sometimes we argue because, you know, in this plot, I was saying we use something really naive, is that here we are actually not even trying to estimate this UKs. We just gave a bunch of possible configurations we can think of from our head. Like, you know, you can just randomly put in a bunch of these numbers in a mixture. But then your assumption is that, you know, whatever component you're putting in there, some of them has to be capturing some true effects. And then because the next step is to estimate these pies, right? So this will be very much looking like the non-parametric approach. You just throw in all the possibilities for this UK, whether or not it's estimate or artificial or whatever, but then you use your data to also estimate the weights there. And if you're doing the right thing, then the relevant patterns of association will be captured and have a higher weight than the ones that are not relevant. And in practice, actually, this number of K in our analysis equals to 1200. So we're putting 1200 different matrices here because we use some of the data-driven ones, and we also try to come up with something that we think reasonable for the context of biology, and we throw in there. But we end up having, you know, non-trivial pies of up to maybe only 20 of them. So we actually remove like over 1,000 of them have a really low estimate for these weights.
Okay, I see. Actually, I have another quick questions about the first part of your talk, the SUSY algorithms.
Yeah.
So you said it was extremely fast. So I'm just wondering if the implementation of this algorithm, does that depend on, for example, the error structure of the regressions, or is it more commonly, it's more generalizable to other regressions or other models?
Great. So the algorithm itself is generic. It doesn't have to assume, it doesn't have to be a sum of single effect regression. It can be a sum of single effect, you know, just a sum of single effect model. So you can imagine putting in, well, so the only structure that has to be a linear structure, the sum is a sum part. And that corresponds to our variational approximation of, you know, factorization here. So this part has to be that. But in here, you know, you don't have to assume xb. You can assume any function. And then as long as the function, so even something nonlinear going on, that's fine. But as long as the function between those functions, it's a sum of effects, then that should work without a problem.
Then what about, then what about the uncertainty quantification, since you're assuming so you're using a variational approximation to the posterior. And you also mentioned like the credible sets at some point. Would that give you the right uncertainty or the right coverage of that credible interval using a variational approximation?
Good. So yes, we can't theoretically guarantee, and actually it's known that you just can't guarantee the coverage, and we can't make a general statement. But at least we can say that in our context, and we did some empirical analysis, that, you know, the coverage is okay. But then still, you know, we can't say it's really, so it's not as tight. So sometimes we aim at a 95% coverage. Most of the time it gives 95%, sometimes it gave only 90% coverage. So the reported, but the results that we report here is actually has additional information saying the actual coverage it attained. So this is one disadvantage of using, you know, this type of approach that doesn't have this guarantee. But, you know, it's a general problem, and I think, you know, advancement in that aspect can be, you know, adapted to our future analysis, hopefully.
Okay, thanks. Is there any other questions for the speaker? Well, if there's no further question, let's thank our speaker again. Thank you very much for a very nice talk.
Thank you.
Thank you.
Well.
All right. See ya.